I'm Michael Collingwood. Thanks for the introduction, of course. Obviously, everyone knows about IDT, or I hear a lot about people knowing about IDT. It seems to be, started out of a small company 25 years ago, and it's just, gotten bigger since then. As I mentioned in the small group sessions, which some of you have been in with me, that this would have been nice to have about 6 months ago when we, first really dove into CRISPR, just to know that the technology existed. A lot of people using talons or ZFNs. We never jumped into that category. It's all protein-based. But with this one, you can use synthetic DNAs or RNAs. So obviously, it really, got us moving in this direction. So to start off, I'm a bench worker. I sit at the bench. I do stuff, at the bench. That's what I do. So with doing this stuff in cells, we run into issues, and the biggest problem being the plasmid-based approach, which is just a really big plasmid. It's 9.5, 10 kb, of a full complete system with your Cas9 protein, on all the promoters, and it's a single guide, one at a time. So for each new guide that you want to look at, you have to make a new plasmid. It can be a lot of hassle. Not to mention hard to get into cells. We've done some electroporation, but a lot of it has just been strictly just liposuction. It's cheaper. It's usually easier. The Amaxis system, you have to get all the equipment, and not to mention the specialized buffers, right, for all the different cell lines. So doing a cheaper approach seems to be nice for our customers, at least as like a starting point to go into. So with having such a big plasmid, you have transfection efficiency problems, which I just mentioned. So you can see that you've got low efficiency on the left-hand side with the GFP tagged, the big Cas9 plasmid. Whereas if you got a smaller 4.5 kb GFP plasmid, you can get much better transfection. And this correlates to on a protein gel, where you can see Cas9 is overexpressed in the middle lanes there. Looking at just the transient transfection in 293 cells with a plasmid, even though you get really low efficiency, like 10% or so, you get huge abundance of Cas9, which may be an indication of off-target effects. We have no indication of that, but more Cas9 you can imagine is going to be detrimental to a cell or off-target effects that you're looking at. So because of this, we decided to just make our own stable line. Those are the 3 lanes actually on the left there. It actually has a single copy of Cas9 integrated through the Droplet Digital PCR system, so we know it has 1 copy per cell. So that's what we've been doing like all of our screening on for now. So as for all these triggers, there's different ways that you can put this stuff in the cells. There's the DNA plasmid, which is U6 driven. You can make a small plasmid that just has your guide and tracer onto it. Put that into either a stable line or whatever. If you're putting in a plasmid or mRNA that has Cas9 on it. Another one is one of our G blocks, which is just basically a PCR fragment, which is the same thing, just U6 driven. SG RNA plus your tracer. You put that in the cells. It makes your RNA. The in vitro transcript, which a lot of people seem to be in favor of. You just make the RNA of your of the complex. The one that we've been heavily towards is the 2-part system, which is the natural system. So you've got your CRISPR RNA and your tracer, which is the picture on the left up there. It's just a 2-part, where you just anneal them together, put them in the cells. We understand the idea of the sgRNA. It's a nice just have a 2-piece system. You've got your protein and you've got your DNA or RNA and put it in the cells. This has 1 extra piece, technically. So going on to that, so we've got on the left-hand side, you've got the natural tracer. So it's got your 20 base target. You have the complementary piece, which is another 22 bases, and then you've got an 89-mer, the tracer RNA that just loops back on itself. So then on the right side, we've got the single guide RNA. They're a little bit longer. They are hard to make, which is why I'm super impressed that Agilent, can make 113-mer sgRNA with super high quality. So that's great. On the other hand, it is hard to make it, and it's hard to make it be clean without it being, cleaned up. So, if we're getting to 89 MERS, we're reaching, or 90 MERS, you're getting to the high end of what can be synthetically made, at least for us in this case. So going on, so the natural choice is to shorten everything. Can we make this thing shorter? In the case of Cas9, people try to shorten Cas9 to make it easier to get into cells. We do the same thing with the 2-part system. So we just, keep chopping and chopping, and at some point it's gonna, not work anymore. But we want to know at what point is that going to happen. So I'm going to mention the T71 assay. This is the most basic kind of assay. It's quick, easy, that people can use to analyze their indel events. You just PCR out the fragment, you treat it with T7, and then you just run it on a gel or some other machine that can give you a readout of the products. So on that third picture, I left in the actual mismatch just because the T7 cleavage assay isn't 100% efficient. It doesn't catch all indel events that we see. So if we go in sequence, we can actually see that there are more indels than what the T7 actually gets. But it does seem to be the best that we have available right now. So once you do your cutting, you're going to have all these 3 species as well as your cut products. We actually use, it's called a fragment analyzer. This is just a really kind of a fancy HPLC where it goes in and it'll give you your full-length product, your 2 little products, plus it'll be quantitative, so you can actually give it a percent, and not to do this image J stuff or whatever with the gel images, which are fuzzy. And luckily, I've never had to use image J, but I know about it. So this thing has actually been really nice to us. It's 96 well based, so you can just throw in a bunch of stuff and let it go. This is just a little schematic of our process of what we do to go through before transfecting and then analyzing all this stuff. So this is just an example of a coworker of mine who she was running gels for a long time, these 2% agarose gels, and just to show you the similarity between what you see for gels versus what we get on the fragment analyzer, as well as the indel percents that it kicks off from there. So after seeing this data, we were more than okay going ahead with the fragment analyzer data. Just to re-mention on the T7 cleavage assay, from what we see for T7 cleavage versus actual Sanger sequencing editing, in some cases we see twice as much editing. So even if we see a 40% cleavage, really the indel event is more like 70%. So, if the indels are mainly one base, you may not catch it with T7, but once again, it's the cheapest and easiest way to do it. So just keep that in mind. So this is just a quick representation of what we've seen for all the different kinds of triggers that you could use, whether it be plasmid, the PCR-based G-block, IVTs, the native 2-part or the other 2-part. We can get good efficiencies with all of these. The 2-part for us seems to be a little bit better, at least our truncated 2-part, the one that we use. And then going along with that, we've got our shortened version. So we just call them short and long versions. So we've got the short one in our stable line. It says HPLC on there just because we tried just a standard desalt tracer. It needs to be cleaned up in our case. We have gotten away with just a standard desalt CRISPR RNA, and those don't seem to be any problems. So you can see the difference between the native CRISPR RNA versus the short 2-part. This is just a 12-site comparison on our standard HPRT1 gene. And you can see the differences in efficiencies going across with both systems. So it's not, not every site is going to be 100% efficiency. And then you can see that in the screens that we have. So we've picked a few different genes, looking at either different exons in that gene, some high, some low GC percentages. And this is just a nice kind of ski slope showing that, if we pick every PAM site in there and make a CRISPR RNA from it, you can get wide variations from it. Some work really well, some don't work really well, and it's not always consistent with certain GC percentages. So like some, high GC percentages, and we can see a good 40%. This guy up here, we've got like 80%. Some, it just depends. So chances are, our moderator actually mentioned earlier, he always orders 4 CRISPR RNAs. Chances are at least 2 of them are going to work. A lot of times 3 or 4 of them work. So right now, we're looking at an SVM algorithm to see if there's any correlation between the 2. But right now, it looks like it might be, it's a good 50-50 chance that whatever you order is going to be correct. These are just the numbers looking at how our high throughput screening has looked in our Cas9 cells, looking at different percentages. If it's got a greater than 15% cleavage or 20% cleavage, we're well up in the 90s, 80s. There's one. Well, the lowest one is 75%. So we've gotten really good efficiencies with our just overall screening of these different genes. And of course, mentioning the RNP complex, which we are big fans of. So making stable lines isn't for everybody, especially for us that we want to screen a bunch of stuff to find out what's going to be best for the people that want to use them. So doing the RNP is nice in a lot of ways because you can put it into virtually any cell type, assuming you can get it into cells. And usually we can get away with a lower concentration. of protein versus the two-part system in like our Cas9 cells. So there's lots of advantages to the RNP concept of this, and this is just a graph showing that versus the RNP and our standard Cas9 cells, we see virtually no difference between efficiencies between the two systems. So with just lipofection, in this case we're using RNA IMAX, in the 293 cells we don't see any difference between the RNP complex and our Cas9 cells. And IVTs are really popular. We seem to have, well, there seems to be issues for us with IVTs. So in this case, we just use chemically or bought products from, in this case, Ambion or NEB. So there's a big difference between the two. As you can see, like in the second slide, we saw a lot of toxicity with the IVTs, whereas with the NEB kit, we didn't see anything. This was even with removing the triphosphate or leaving it on, we still saw the difference between the two. We have collaborators that love IVTs. And that's great, but overall, and like we don't see any toxicity. So obviously we switched over to the NEB kit. So then looking at the IVT versus the two-part stuff in just our Cas9 cells, we can see that in a lot of cases, we see similar efficiencies, but there are a few cases where the efficiency, has dropped, or you don't see it at all, depending, like maybe if the CRISPR RNA wasn't as good to begin with, or a couple cases where there's a huge decrease in efficiency. And because they're IVTs, even if they're FOST, a lot of people don't look at interferon responses or to see what else is happening in cells. So this is just an example of a couple genes that we looked at that are induced for interferon responses. So the orange bars are the IVT, the blue ones, which you can't really see, and there's a reason for that because of the fold changes here. But they are there, there's nothing happening. So IVTs are causing some kind of effect, but a lot of people don't look at this. Kind of like off-target effects, if you don't look for it, you're not gonna see it. But just to keep that in mind that other things are happening in the cell even if you don't recognize it right away. And just to show, we've scaled up and scaled down for nanomolar amounts, so in case maybe 30 nanomolar isn't ideal for IVTs. We've gone up to 100 nanomolar, we haven't seen really any benefits. We've gone down to 10 once again, maybe to help reduce the off-target or the interferon response, and don't really see anything for that end. 30 nanomolar seems to be the ideal for the two-part and what we can get for the IVTs. Also time course, thinking that the RNP would be easier, and maybe get your results faster after 24 hours. That doesn't seem to be the case. It still, at least in our case, takes 48 hours before we still see the maximum amount of efficiency. And that could just be because the NHEJ it's efficient at repairing it the correct way that you need it. More time to just keep cutting and keep cutting and keep cutting for it to give you the indel that you want to see. So then looking at the two-part IVT and the RNAs in the RNP complex. So this one took a much bigger hit than just our Cas9 stable lines. So doing the IVT with the RNP, I mean, we're down into very low levels than what we typically see. So this one didn't correlate as well going from the Cas9 line into the RNP. So in conclusion, we obviously like the synthetic RNAs. So the two-part system, it mimics naturally like what happens in the cells. So that's why we're kind of geared toward that way. The RNP obviously works really well. We have a semi-optimized shortened CRISPR trace RNA, which seems to work better than the native system. We're not sure why that is, or we can go shorter, or maybe make things a little bit differently. Slightly better, performance is slightly better than the expressed sgRNA, at least in our hands. The IVTs don't seem to work as well. Our collaborators love them, so everyone likes what they like. The two-part system shows a high positive hit rate in our walks that we've done. 5 genes, 6 exons or so that we looked at. So we're looking at the algorithm. At the same time, we don't know if it's really going to impact anything or the way people order stuff in particular. So everyone has their shout out. Sorry, I don't have that many people on here, but our lab isn't that big. So the main person that's been working on all this stuff is pretty much everyone here except for Mark, of course. He's the big guy that sits in the office. So Ashley, Molly, Garrett, and Sarah. I mean, we've all been working on this stuff a lot, and it's been a very big learning curve since December that we looked into it. So maybe next time you guys could do this a little bit sooner. And with that, I'll be ready if you guys have questions or my email address, email me if you guys have questions. We're more than happy to answer anything, or, tell us our stuff, great, sucks, whatever, If you guys are interested in trying this stuff, we'd be more than happy to send you stuff, or if you guys have IVTs at work, we'll take them and try them and, we'll see what works for us. But thanks.